All right, this is the lecture for chapter 20. We are starting now the material that's going to be on test number 3. We're going to do a quick survey of the bacteria, archaeans, viruses, which is a special interest right now. Then we're going to go to protists, fungi, and plants, and all that will be on test 3. So, evolution of a disease. This is really critical right now. We're in the midst of a pandemic. So, bacteria and archaea are single-celled organisms without a nucleus. Remember, prokaryotes. But look at viruses, even simpler than bacteria. They have no chromosomes, no metabolic structures. They can't do anything on their own. They don't use energy. They don't make energy. They have no organelles. They do not do anything. They're not alive by some definitions. And for most people, they would say they're not alive. Remember, as we started off, we said there was 3 different characteristics of everything that is alive. 1, they need a constant flow of energy. Well, these don't. Number 2, they respond to change. Does an individual virus respond to change? No, but collectively, they do, in a way. They adapt and mutate. The third thing was, do they have DNA? Well, most viruses don't even have DNA. Most viruses are RNA. COVID-19 that we're dealing with right now is a RNA virus. So that's where we are. So they're not alive by what we talked about at first, although most cellular biologists will say that it is a life form. It's just different than what we classify as a life form in biology. They can evolve because they have a genome that can mutate. It's a very simple genome. It's no chromosomes, just usually a strand of RNA or a strand of DNA. And we'll talk about that here in a little bit, how they get into our cells and how they replicate and what they do. So, HIV virus, the cause of AIDS in humans, was first isolated in the early 1980s. The HIV-1 is the most common strain and evolved from a type of SIV that infects chimpanzees. It mutated and affected a human who hunted and killed chimpanzees for food. What kind of person does that? But that's how this disease came into humans. Somebody killed a chimpanzee and ate it, and so the virus came in them. In 1959 was the earliest evidence of HIV in humans. The virus probably made the jump in the early 1900s. So this happened a while ago, but it was identified in the 1980s, and we're still dealing with this virus today. Gene sequence comparisons allow researchers to track movement of HIV out of Africa, so they were able to track this by looking at the genome of it. The HIV virus is carried to Haiti in the mid-1960s. It mutated and diversified in Haiti, traveled to the United States by 1969, spread quietly until identified as a threat in 1981. 30 million people are now infected with HIV today. So this is a virus that once it gets in, you cannot get rid of it. You don't develop an immunity to it. And we'll talk about that as we go through this chapter too. So what's a virus? It's a non-cellular, it's not a cell, infectious particle consisting of a nucleic acid and a protein. That's it. Remember, what's the smallest unit that can exist as life? It's the cell. It's the smallest unit of life. This thing is not even a cell. So in most definitions, it's not alive. A virus is smaller than any cell, has no metabolic machinery of its own, it has no ribosomes, it can't make proteins, it has no other metabolic machinery, and it can't make ATP. It doesn't use energy, it can't make energy. A virus can replicate only in a living cell. So a way to think about a virus is it is a microscopic parasite that gets into our bodies. It does act as a parasite. The virus must insert its genetic material into a cell of a specific host. Some viruses are disease-causing agents, which is pathogens. Notice for the test, no definition of a virus. It's a nucleic acid in a protein, no metabolic activities, and a disease-causing agent is a pathogen. Viruses, bacteria, some fungi, cause diseases. Now, before I go further in this, I always ask my lecture class, do you believe in complete evil, total evil? Do you believe that there's something that's just evil? So, do you believe that something is evil? And so the closest thing that I can find to something that is just evil are these viruses. And we're experiencing it now, right? In the middle of this pandemic. We've been shut down now for 3 months? Stay-at-home orders. We're opening up now finally, thank goodness. I don't think we needed to close anyway. We're going to talk about how viruses run as we go through this too. Our best hope is to get a herd immunity. As more and more people catch this, it just won't travel quickly and it won't spread near as much if it does pop up. So, all right, so what is the viral structure? It has a viral particle, a viron. It includes a viral genome enclosed within a protein shell, the capsid. So basically, it's just a strain, most of them are a strain of RNA wrapped in a protein, or a strain of DNA wrapped in a protein. COVID-19 that we're dealing with right now is an RNA virus. So it's a strand of RNA with a protein sheath around it, and this particular one, since it's an animal virus, is also enclosed in a plasma membrane. We'll talk about that in a little bit. So the genome may be RNA or DNA. COVID-19 is RNA, single-stranded or double-stranded. So if it's RNA, it's single-stranded. It has to be converted into DNA as it gets into our cells. The capsid consists of a protein subunits in a repeating pattern, producing either a helical, which is a spiral, or a polyhedral shape. COVID-19 is polyhedral. Some components of the viral coat bind to proteins at the surface of a host cell. Remember when we talked about cells, that plasma membrane, and there's these receptor proteins that stick out. These viruses, when they come to those receptor proteins, the plasma membrane that is encircling the virus is yours. The virus gets into your cells, replicates, comes out of your cell, and takes part of your plasma membrane. So when it gets to a new cell, your cell identifies it and says, hey, that's me, because it's got my plasma membrane around it. So some components of the viral coat bind to proteins at the surface of the host cell. All right, 4 characteristics of a virus. Know these for test 3. They're non-cellular, they have no cytoplasm, no ribosomes, no typical cell components. The genetic material is either DNA or RNA. They can only replicate inside of another cell. They're parasitic in that way. And they're very, very small. We can only see viruses with an electron microscope. So helical viruses that infect plants, like the tobacco mosaic virus. There's complex bacteriophage. Now, what's a bacteriophage? That's a good question for a test. It's there in red, right? This is a virus that attacks bacteria. There's a whole classification of viruses that just attack single-cell bacteria. There's polyhedral naked viruses. So example of that is the adrenoviruses. And there's enveloped viruses, herpes viruses is an example of that. COVID-19, which this book was put out before this hit, COVID-19 is an example of an enveloped virus. All right, so here is the tobacco mosaic virus. This is a plant virus. Most plant viruses, we said, is that helical structure. Here's the proteins going around and around and around, and there's our strand of RNA that's sticking out of the top. And when this infects a plant, it doesn't kill the plant. You know, parasites, good parasites, never kill the host. They just use them. And so this is, you know, the leaves wither, get brown, and so it's just not good anymore for the use that humans have for it, which is probably good for tobacco. But anyway, so it's not usable. It's a disease. This is a bacteriophage. I love this one. This one reminds me of the lunar lander. See, there's the little arms at the bottom of it so that it lands. Here's the capsule up here, but inside of this there's a DNA. This is the protein coat. And this thing actually has a little needle-like projection that it will stick. Here's actually one under an electron microscope on a bacteria. It'll actually stick through the plasma membrane or the cell wall of the bacteria. And then a little needle-like thing will come out and inject the DNA inside of the bacteria. So that's a pretty cool looking virus, but that thing's not alive. It has no neurological functions. How does it know to do what it does? It just doesn't. Again, that's the way I define it. Here is an adrenovirus. He's got that polyhedral shape, some protein sticking out. Here's one that's enveloped. So this is now enveloped by a plasma membrane. And there's what it actually looks like under a microscope. The virus is here in the middle. It's covered by that plasma membrane that it stole from your cell as it exited out of it. So your body views that as me. All right, an adrenovirus. They're actually viruses are pretty cool looking in a way. Very artistic looking, I guess. There it is with the envelope around it. The protein coat. You can see it beneath the envelope. But these are the recognition proteins that it took from the host. So this is the plasma membrane, all these proteins sticking out. So there's the closer view of what it looks like in the microscope. All right, viruses infect and replicate in all organisms. We just learned bacteriophages. They're in bacteria. Plants, we saw the tobacco mosaic virus. There's viruses in all plants. They usually enter a plant when the plant has some sort of damage to it. An insect has bored a hole through it, or a branch has been knocked off, or whatever it is. There's an opening, the virus invades. So all organisms, animals, everything can get a virus. Viral infections often decrease the host's ability to survive and reproduce, and affects many ecological interactions. Humans benefit when viruses kill pests that feed on crop species, or when bacteriophages kill toxic bacteria. Now, we say humans benefit. This is a virus. What do viruses do? They mutate, and they mutate quickly. And so, there you go. We bring a virus in to kill a pest, and what can happen? These things are going to mutate, and they're going to start going after other things. And so, don't play with viruses. Governments, don't do it. This one we think started in a viral laboratory in Wuhan, China, because they just made a mistake. They were studying these viruses, but man, this is something we shouldn't be fooling around with. Viruses are dangerous. We say they benefit. Man, limited benefits but you're playing with fire when you're trying to get a benefit out of a virus. Viruses harm us when they infect livestock or crops that we use, and they can cause human diseases. Yeah, no kidding. With COVID-19, we've got this upper respiratory infection, and it's really not the virus itself that's killing people, it's our response to the virus. Our immune system just flares up so radically that it actually causes damage to our own tissue. There's diseases in humans that do that, like lupus, where the immune system is just starting to attack its own tissue. So these viruses, they're bad. Now, why are they bad? Well, let's go on here. There are viroids. They're small RNAs that cause disease in many commercially valuable plants. They're small, circular, single-stranded RNAs with fewer than 400 nucleotides. That is really small, by the way. They do not have that protein around them, so it's not even a virus. This is something smaller than a virus. It really doesn't affect animals as much as it does plants. The virus is replicated in the plant cell nucleus by the plant's RNA polymerase. We'll talk about that when we get into animals. RNA viruses. All right, so how do these things replicate? There's not male or female. There's no metabolic machinery. They don't split by binary fissions. How do they replicate? It's like cellular hijacking. Now look at that. A viral infection is like a cellular hijacking. What's that mean? When somebody hijacks, a person hijacks a plane, what do? They go up to the pilot, they put a gun to his head, and they say, "Fly me here and nowhere else." That's hijacking. The virus will enter into your cell and enter your nucleus and tell your nucleus to make nothing else but me. It completely takes over your cells. So it takes over a host cell machinery and directs it to synthesize viral components that can self-assemble as new viral particles. So every time now that RNA comes in to make a copy of your DNA, it's going to be directed to this new viral part. So we'll make this a little bit more simpler here. The virus enters your body. It's RNA. It enters your nucleus, and through reverse transcriptase, it becomes DNA. That DNA then embeds itself in your DNA, takes over your nucleus. So now that when new copies are made, it's going to make copies of that viral. And so that virus is then going to be taken out, and the proteins are going to be, the code for that protein that's going to surround that new virus is made in the ribosomes. The RNA is copied and it's put out. And so we've got your cell making the proteins for new viruses, RNA for new viruses, and then when they touch inside the cell, they self-assemble into a new virus, and then they will leave your cell by pinching on part of your plasma membrane and go and do this to another cell. We'll take it and hijack it and take over. So, yeah, this is, you're starting to see what I meant when I said evil. They can't do anything on their own. They can only take over your cell stuff. So there's steps in viral replication. We talk about 5 of them. Attachments. Proteins of viral particle chemically recognize and lock into specific receptors at the host cell. For these enveloped viruses like COVID-19, HIV, it's got your plasma membrane, so it naturally will stick to your cell. The protein will say, okay, that's me, will come on in, and it'll bring it on inside. So that's attachment. Penetration. Either the viral particle or its genetic material crosses the plasma membrane of the host cell and enters into the cytoplasm. We're going to see some examples of that. Replication and synthesis. Viral DNA or RNA directs the host to make viral nucleic acid. So if it's DNA, it'll make new viral DNA. If it's RNA, it'll make new viral RNA. And then those proteins that will wrap around the new virus. Assembly. This is just amazing. They self-assemble as new viral. How do they know to do this? Whenever they touch, they will self-assemble. I don't know how. Release. The new viral particles are released from the cell. So know these steps of viral replication. This is a topic we should be really looking at. So there's two pathways that viruses take in a host body. The lytic pathway, under direction of the viral genes, the host makes an enzyme that lyses and kills the cell. So basically, there's so many viruses that build up inside the cell that it then says, okay, let's burst this cell open and release all these viruses. So that's one way. The lysogenic pathway, the virus enters a latent stage. The host cell replicates. So let's say this is a bacteria. The virus enters that bacteria. You know anything about bacteria, which we're going to learn in this chapter, they divide really rapidly. So it enters into the cell and it goes latent. The bacteria divides. Now you got two bacteria with that virus in it, the viral DNA or RNA. Then they replicate again. Now you got 4, they replicate again. Now you got 8, they replicate again. Now you got 16, they replicate again. Now you got 32, and it goes on and on and on until something wakes up the virus. It takes over all those cells and then start pumping out viruses. Sometimes when the reason why the flu can hit us so fast and so hard is that it has entered the lysogenic pathway in our cells. And so those viruses just start, when they wake up, your cells have multiplied. And now there's just a lot more of it in there. And then once they come out in mass numbers and they can hit you just like that. That's some, of the sicknesses we get. That's why they can hit so fast. Others are just a slow build-up. The way most viruses work, it's a slow build-up. And COVID-19, 14 days is what they're saying now. All right, this is a bacteriophage. So here's our little lunar lander. It attaches itself, and it then inserts its little genetic material. That drawing reminds me of a fish swimming and something coming out of the fish, but it inserts into the bacteria. The host replicates, and so it makes new proteins and new RNA or DNA, and then they self-assemble inside. Then the accessory parts are attached to the viral coat. How? It's all metabolic activities, chemical signals, enzymes, but yeah, this virus is taking over the cell. It's only going to make it now. Until finally there's enough of them in there that they lysis, they break apart the cell wall and plasma membrane of the bacteria, and now there's a bunch of them released all at once. So that is the lytic pathway. Well, there's another pathway, that lysogenic. So here, the DNA or RNA is inserted, and look what it does. It places itself right inside of the DNA of the host. Now, if this was RNA that comes in, it first has to go through reverse transcriptase here. There's a lot more probability of getting a mutation through reverse transcriptase. So RNA viruses change much more quickly. Remember, COVID-19 is an RNA virus. This thing is going to start mutating. It already has. There are different strains of COVID-19 already. It's going to keep mutating and changing. Chromosomes and integral RNA DNA are replicated, so it wakes up, starts making more, the cell combines. Okay, I'm sorry, it hasn't woken up yet. This is just the cell duplicating, making more of them, and then it gets to this point where they now wake up and start producing the proteins and more RNA for this thing. It will then enter the lytic pathway until it breaks open the cell wall and plasma membrane, and they come out. All right, replication of HIV. Why is HIV such a nasty little virus? We'll see here. So HIV binds to proteins on the host cell. The viral envelope fuses with the host membrane. The viral RNA enters the host cytoplasm. An enzyme, reverse transcriptase, there it is, converts the viral RNA to DNA. That's what's happening with COVID in us too, which integrates with the host DNA. Host cell produces viral RNA and proteins which assemble into new viral particles. The new viruses are enveloped in the host plasma membrane and exit by exocytosis. There you go. So here is HIV. So this is a plasma membrane of its host that's surrounded. Here's the proteins embedded in the plasma membrane. Here is the protein sheath around the HIV virus. And this is one of the two strands of viral RNA that's in HIV. All right. So here it's coming. So this is the virus. It's covered with your plasma membrane and proteins. So your proteins on the new cell, typically white blood cells, read these and say, hey, that's us. This is a friend. Come on in. And so look what happens. It comes as part of the plasma membrane. It injects the RNA inside. What does the RNA do? Well, let's go through the steps here. There it is attaching. There's the RNA that comes through. Reverse transcriptase now makes it into new DNA. The DNA is then inserted into the DNA of the host cell. Now, bad news. Once this happens, it's in there. That's why there's no cure for HIV. It's now a part of your DNA. If you want to go in there and get rid of that, you've got to destroy all the DNA, which is your cell can't survive then. So once it's in your DNA, it's there. And now guess what's going to happen? Transcription to make new RNA, and it's going to take over also to make the new proteins, and they will self-assemble. They will come to the plasma membrane, and then it pitches off by, it take part of the plasma membrane of the host cell with it as it leaves, and it becomes a new virus to go to the next cell it's going to attack. All right. So viruses as human pathogens. Well, this should be interesting right now, right? Viruses, bacteria, fungi, and protists can cause human diseases. Well, of course we know that, right? Viruses, what we're dealing with now, the common cold is a virus. Influenza every year is a virus. Bacteria can cause different diseases. Fungi cause different diseases. Athlete's foot, we've got different things fungus cause. Protists, we'll talk about, and we go through the chapter on the protista. They can cause, many of them act as parasites or can cause diseases. Viral diseases range from merely inconvenient, common cold, to potentially deadly. COVID-19. So this is a very depressing slide. Here is the disease, the pathogen, how many deaths per year worldwide. Acute respiratory infections caused by viruses and bacteria. COVID-19 would be included in this first group. 4 million. Now this year it's going to be a lot more 4 million because we've got this pandemic going on. But that's the average, 4 million. AIDS, HIV virus. 2.7 million people die every year from AIDS. Now, how to phrase this? Yes, every life is unique, it's valuable, has purpose. I'm not arguing that. From my worldview, we have, we are created with purpose. And so every life is valuable. My worldview says that our number one command is to love each other. So every life is valuable. Look at how many people these diseases are killing. And this is just mind-boggling to think. On the average year, 4 million people die from these respiratory viruses. That's just mind-boggling. Right now, in the United States of America. With COVID-19, we're hitting 100,000. Now, I don't want to say that's great, but what we were looking at when this started, there were some estimates up to 1 million, 2 million people just here in the United States. Well, so, this pandemic is bad. Thankfully, it's not near as bad as first feared. I can't imagine if what happened today would happen back in the late 1800s when we had, a million people die. So, this is bad. But, worldwide, that's just a mind-boggling number. Diarrheas caused by viruses, bacteria, protists. 1.8 million people die every year of diarrheas. Dysentery, especially in countries where there's not clean water. This is where it really hits. You start to get bacteria or protists growing in the water, and it can cause that. Tuberculosis is by bacteria, 1.6 million. Malaria, which is a protist, 1.3 million. Whooping cough, 294,000. Tetanus, 204,000. Meningitis, 173,000. Syphilis, 157,000. So, these things cause a lot of damage, and there's no doubting that. Look at what's going on right now in our country. These things cause a lot of damage. All right, so an infectious disease arises when the activities of the infection agent interfere with the host's normal functions. Most infectious diseases are spread by contact with mucus, there you go, blood, or other body fluids that contain the pathogen. That's why with this COVID-19, we've been saying, wash your hands. You come into contact with it. You touch a surface that has the virus on it, where somebody has just gone by, they've sneezed or coughed, and the virus is on. You touch it. For most flus, this is how it's transmitted. You touch something, you then touch your eyes, your nose, your mouth, and now that virus is in your system and will start replicating. For COVID-19, they were saying, and still are saying, wash your hands all the time. That's why. Use soap and water, hot water, because the heat will help to denature that protein. The protein will start to unfold. The detergent will break the hydrogen bonds of the protein too, and it will start to unravel, and so it can't function. And remember, proteins only function if they can hold their shape. And then, the soap can also kill the, if it's enveloped the plasma membrane, oil and soap, soap breaks apart those lipids, and so it will destroy that membrane around the virus. And so, washing your hands does a lot of good. But also, this is spread by, droplets in the air, microscopic droplets where we cough or sneeze, or even when we talk, we are, when you say a word of a P in it, and there's a little bit of, and so that's why they're saying 6 feet is the typical range of that. Now, a sneeze will travel farther, but hopefully people learn now that you never just sneeze towards somebody. You cover up and do all that. Other infectious diseases require a vector, such as a biting insect that carries the pathogen from host to host. Give you an example of this that we're dealing with also right now in West Virginia, Lyme disease. Where does it come from? It comes from a little teeny tick that will get on you, inject you with this virus, and now you got it. And so, all right, let's go on. We're going to talk a little bit more about COVID-19 here in a minute. Most viral diseases cause mild temporary symptoms. Upper respiratory infections and common colds, and intestinal infections that cause vomiting and diarrhea. So those are normal things that viruses cause. This upper respiratory, though, you like the flu, when people catch the flu, it's not even the virus that really kills people. It's the virus, it's an upper respiratory, and that will quickly turn into pneumonia. And so the pneumonia is what's going to be killing most people with the flu. Some viruses remain in the body in a latent state, and they can reawaken later on. Cold sores. is herpes. So they will stay latent and then they will fire up. And you'll get a cold sore. Genital herpes, another form of herpes, latent for a long time, then it will fire up and you become contagious. Mononucleosis, HIV. Some viral diseases can be prevented by vaccines: measles, mumps, rubella, chickenpox. This chickenpox is one that stays in you. Once you have chickenpox, that virus stays in you, but it's latent. For some reason, it wakes up. Most theories are under times of intense stress and pressure. And when chickenpox comes back, it doesn't come back as chickenpox, it comes back as shingles. I dealt with that when I was getting my master's degree at Marshall University. I had shingles. Not a nice thing to go through, very painful. And there's nothing you can do about it. Basically, it just runs its course and you have to deal with it. So some of these things can stay latent. Now there is a vaccine for chickenpox. Good thing to have. The chickenpox itself is really not that big of a deal. Shingles is a worse deal than the initial disease. Most viral diseases produce mild symptoms. Symptoms usually subside quickly. Some viruses can remain in the body in that latent state. There's herpes simplex. There's the virus that forms for chickenpox, comes back as shingles. Some viral infections increase the risk of cancer, like the HPV virus, the human papillomavirus, can actually cause cervical cancer. So there's vaccines for this now. So all right, we're going to pause here, and we'll go up just a little bit more. All right, West Nile disease was first identified in humans in 1981. It spread to North America by 1999. Yay, our lifetime. 5,000 cases in the U.S. in 2012. It's transmitted by mosquitoes, another vector, mosquitoes. Mosquitoes can give us several different diseases. Ebola virus, now that's a nasty one. You think COVID's bad? Ebola. What is the death rate of COVID-19? The CDC put out some information this week that the best guess that by the time that everybody is tested, it's going to be about 0.3% mortality rate. So if you catch COVID-19, what's that mean? You have a 99.3% chance of living through this, okay? For the average person, 99.3% chance. I mean, that's great odds. If you would make that on a test every time you say, yeah, I'd take that, a 99.3%. So this is not that deadly of a virus. Now, the common flu, that the yearly flu, is about 0.15. So this is twice as deadly as the flu. Now, the bad thing about COVID-19 is it's twice as deadly, yes, but it's also much more contagious. It spreads more quickly. Part of the reason is it's a novel virus, so we don't have the ability to recognize it and fight it yet. And we'll talk about that here in a minute too. So the Ebola virus, talking about, COVID, 0.3, that's 0.3 of 1% of people who catch it die. Ebola, people who catch it, it's over 60% of the people who catch it die. Now that is just astounding. So if that came to America and just spread like wildfire, that's half of our population, well over, 150 million dead. So, not good. So this one was really bad. First isolated in 1976. It's extremely contagious in humans. Outbreaks have been limited to Africa to date. Why are outbreaks limited? Because most people who get it die. And so you're not walking around spreading it. When you get Ebola, this is not something like COVID. The amazing thing about COVID is this virus that is, it's almost like the perfect little virus for a virus to be. 60 to 80% of the people who catch COVID-19 virus, they don't show any symptoms. They don't even know they're sick, or they have, I just got allergies going on, or I got a mild cold going on right now. So Most people don't even know they have it. When you catch Ebola, man, you know you've got it because you're almost dead at this point. And so, yeah, it doesn't spread. There's not people walking around saying, yeah, I'm healthy and I can go be with people, when in fact they're carrying a virus. That's why COVID-19 is spreading so rapidly, because if there's that many people who have it and they don't even know they have it, yeah, you're going to go see people. You're going to touch people. You're going to, be close to them. And so, yeah, it's going to spread, and that's what's happened with this pandemic. Ebola, not, and thankfully it hasn't spread. This one, you think COVID's bad, this one is nasty. It actually causes massive bleeding inside, and it starts to liquefy your cells, and it's just a nasty virus. Virus genomes can be altered by mutation. HIV and influenza mutate especially quickly. Remember, RNA viruses mutate quicker than DNA viruses. So there's a new flu shot vaccine created every year to keep up with mutations. Now let's pause here for a minute. Let's talk about COVID-19. Okay, this thing is an RNA virus. It mutates. There's a lot of talk in the news right now about a magic bullet that will help us. We just got to wait until we get a vaccine, and the vaccine is going to save us. I don't want to spread any fear, but you need to know these things. This COVID-19 is going to mutate. If they come up with a virus this year, it's not going to work next year. Every year they have to come up with a new virus vaccine. And what they're doing is, when they make these new vaccines, is they are guessing what the mutation is going to be. Now, it's a scientific guess. They are looking at how these viruses are changing, and they make the vaccines from the little pieces of protein that stick out of that envelope. But if you can recognize the protein, then your cells can recognize this as an enemy and can attack it so it doesn't spread through your body. More, these viral medicines, which I think is going to be the best way to combat COVID-19. The vaccine will work if it pops back this year, but it'll change by the time we get the next year. But these viral medications, there's some of them that, where the virus comes with your plasma membrane with the proteins, and it comes to stick to your cell. There's medicines that will keep that from attaching to your cell to get injected inside. There's other medicines, and this is where HIV has really paved the way. There's medicines that can stop reverse transcriptase so that it can't turn into DNA and get into your cells. There's other medicines that will keep that self-assembly from happening. So there's all kinds of different ways that medicines can attack the life cycle of a virus. And so this is good. And so, yeah, these medications that we're getting, hopefully we're going to start finding some medications that work well with this. Now, what do we just say? Well, we can come up with a vaccine for this. We've never come up with a vaccine for this type of virus ever. If we come up with one, then yeah, it's going to work for a while. This thing's going to mutate. Good news. When these viruses mutate, they normally get less lethal. A good parasite never wants to kill its host, ever, because you want to keep that host alive and keep making you, and so, or feeding off of you, like a tick. A tick is not enough to kill you, but if you get a ton of them, I guess it could, but they don't want to kill the host because the host is what's feeding them. And so viruses act sort of like parasites in that way. So they usually mutate down, and that's natural selection. And so, yeah, it's better for them to mutate down in severity. But you just never know what the mutation is going to be. It can mutate worse. So that's how we're getting some of these bad diseases popping up all of a sudden. These things are mutating. COVID-19 was a mutation, probably between a virus and the person who was exposed to it in that lab, and the one that came from a bat. And so now they have combined together to form this new nasty little virus, COVID-19, that spread human to human, and what's happened. So The vaccine may not be the answer. We may have to come up with a brand new one every year with this, if we're going to include this in the vaccine regimen. A better alternative is, I mentioned earlier, herd immunity. Once, how does the virus spread? People get infected, and then they spray it out, right? As you come into contact with people or things, you're spreading the virus. Once you get most viruses, you have the antigens for that virus, you can't catch it again. And so once your body fights it off and kills it, you have the antigens in there, so you can't get it back. So if somebody comes to me, I've already had it, I can't catch it again. So I can't spread it on to the next person. Once we get about 60% of our population who have had this thing is not going to spread very much at all. So that is herd immunity. Once you get enough of the population with the antigens, then it's much easier to control this. This is a novel virus. That's why this one's so bad. There was nobody in our populations who had the antigens for this. So this was new, and it spread quickly. I hear a lot of doom and gloom talk about it's going to come back next year. No, we've got a bunch of our population that has antigens for this now. We can start to fight this thing off. So it's going to get better. Just stay with it. Keep washing your hands. Wear a mask when you're out in public. Don't touch your face. All those things you've been hearing again and again and again. Yeah, do those. And it can help you from catching this virus. So thankfully here in West Virginia, we haven't had that big of a deal with it. I keep talking with the folks at our local Kroger here. And they have been working every single day almost, way overtime hours. But nobody has caught this there because they're taking the precautions. They're with people every day. You know that virus has been through that store. But they're taking the precautions. They're washing their hands, wearing masks, not touching their eyes, not touching their mouths and nose, and so they are staying safe. So do that. Heard one doctor said, become a face Nazi. Don't ever touch your own face when you're out in public. Now, when you're at your house and you're clean, you can do that. But all right, so viral reassortment. That's probably where this thing started. Swapping of genes between viruses that infect the host at the same time. So this caused H1N1 in 2009. It caused the epidemic in Mexico. Thankfully, it didn't spread too much from there. All right, H5N1. Now, first of all, what are these numbers? H and N. There's two different types of protein that make the viral sheaths around the RNA or the DNA. And one of them starts with an H, one of them starts with an N. So it's just what type of protein this is. So this is how we make the vaccines. We recognize the type of the protein that's surrounding the virus, and we put antigens in the body for those particular proteins. So now when your body says, hey, we got an antigen in that protein, that's an enemy, let's destroy it. And so it keeps you from getting the virus. H5N1 this is the bird flu, an influenza in birds that can infect people who come in contact with infected birds. High death rate in humans. Again, about 60% fatality. That's bad. Person-to-person transmission is rare. Now, that's great. It doesn't spread person to person. It's only from birds to humans, and a lot of times bird droppings to humans. But here's the fear. What if this mutates so that it can start to spread person to person? This is a pandemic we don't want to see with a 60% fatality rate. But chances are, when it mutates to go person to person, it's also going to mutate down in lethality. So that's what these things do. So the coexistence of H5N1 and H1N1, the swine flu and the bird flu, is dangerous because of the potential for gene exchange between the viruses. So if I have the virus for H5N1, and then I catch the virus for H1N1, so now I've got two viruses. Think about what they're doing in your cells. They're hijacking your cells, causing them to make those proteins in the viral, and when they self-assemble, they can grab parts of each other and make something that is brand new. So that's where the danger is here. Possible result, easily transmitted deadly virus. So if we don't want to get these two together, hopefully nobody's experimenting with that. There's some things that no government should ever fool with. And these biological weapons like this is something they should never even think about fooling with. Why make it? Because you would never release it, because you can't control them. They're going to kill you and your people if you release them. You can't contain these things. There's herpes simplex infection, there's a cold sore, and a pleasant picture. All right. RNA viruses such as HIV and influenza, they mutate especially quickly. COVID's in that category. Replication errors made by viral reverse transcriptase remain uncorrected because the host proofreading repair mechanisms do not operate during reverse transcriptase. They don't check to make sure there's no mistakes. Your cell, when you're doing the DNA to make RNA, it's checking every single time because you got to make that protein just right. But when it's going the opposite direction, from RNA to DNA, there's no proofreading. So there's a lot of mistakes can happen. So to keep up with ongoing mutations in the flu, scientists create a new flu shot every year. Isn't that a pleasant thought with COVID-19? All right, so here's the influenza virus. Here is that first protein. There's our H, hemagglutinin, and here is our N, neuraminidase. So that's the two proteins, and depending on which type of protein that is, they will give these different numbers, and so they can be a lot of different combinations. Novel genomes. Have you heard them say that coronavirus is a novel virus? This is a novel genome. Novel genomes arise from viral reassortment, the swapping of genes between viruses that infect the host at the same time. That's what I was talking about. You got two of them in your cells, and when they self-assemble, they will grab pieces of each other and create something new. H1N1 influenza has a composite genome with genes from a human flu virus, bird flu viruses, and two swine flu viruses. So, a lot of different stuff going on to make that beauty. Novel viruses can cause an epidemic. Notice on the test, what's an epidemic? It's an outbreak in a limited area. You should know this word now. Pandemic. Pan is Latin for the world. So it is a worldwide epidemic. This is a worldwide epidemic. It's everywhere. So here's two viruses come in, and look as they come out, and as this new one has come out, it's got one little piece of the other one. So now this is a brand new virus. It'll do brand new things. It affects us differently. COVID-19 has hit when we've got two strains of flu. We had this year flu A and flu B. Our flu shots, remember I told you they make a scientific guess as how these things are mutating. This year's flu shot was not great. At best, 50% of the people that got it kept on getting it. So there was a lot of people out there with flu A or flu B or both. There was a young lady I know who was sick when it got tested. She had both flu A and flu B at the same time. Now, you got two viruses in there. Why does she caught COVID at the same time? Well, now you got three. Now you got the possibility of even more assortment going on at the same time. So this is how we get these novel viruses. COVID-19 was a novel virus. All right, enough of viruses. But you can see why I say, to me, I haven't found anything on the face of this planet that I would call pure evil like viruses. I mean, they just hijack your cells, they take over. They can't do anything on their own. Their only way they can get things done is to bully your cells into getting things done for them. And then they will replicate until they can either just come out of your cells or burst your cells open. And so this thing is, viruses are evil. Well, let's move on. The prokaryotes. Now we're into cells. They either have a nucleus or no nucleus. Prokaryotes, no nucleus, but they do have DNA. They have a single chromosome ring there. They're simple cells that lack a nucleus. They've evolved before eukaryotes. Prokaryotes still exist in enormous numbers. You have trillions of them in your gut helping you digest food. They're there and you need them. They show great metabolic diversity. What's that mean? They can live everywhere. They can feed in any number of ways. Cells without a nucleus are prokaryotes. Those with a nucleus are eukaryotes. That's a repeat of stuff we had in the test before. That's a great questions for this test. I love these things where you can just keep repeating and learning, right? The prokaryotes includes two distinct lineages, bacteria and archaea. So here, this is what, when I was in college, that's been a while ago, we had two domains, prokaryotes, eukaryotes. Now we got three since the early 80s, bacteria, archaea, and eukaryotes. Maybe you know those domains? All right. In eukaryotes, a species is defined on the ability of its members to mate and produce fertile offspring. Remember that from chapter 1. What is a species? They can mate and make fertile offspring. That's not a good definition for bacteria. Does not apply to those that reproduce asexual. So there's not a male and female with egg and sperm making a young. Bacteria just divide in two to create two that were identical to the parent. So you have a mother cell that divides into two, you have two daughter cells. Prokaryotic species are defined as groups of individuals that share an ancestor and have a high degree of similarity in independently inherited traits. How many species of bacteria are there? Nobody knows. There's a bunch. So here's a bacteria. There's the pili sticking off of it, and you can see that it's starting to divide here and to make two identical cells, clones, if you will. So, the field of metagenomics assesses microbial diversity. How do we know the species of bacterial? We're getting to be where we're reading the genomes, the DNA of the samples collected. We had the Human Genome Project. I'm sorry. We did have that. We know the entire human genome now. We know where every gene is. We don't know what all of them do, but we know where every gene is. And the results of the Human Genome Project were just amazing. But there is the Microbiome Project, which studies the DNA of the bacteria inside of us. And so it's a study of microorganisms that live in or on the human body. So there's more than 1,000 species of bacteria and a few archaea species that live in the human gut. So you got all these in there. They're doing good things. Don't be grossed out. You have bacteria over your skin. They're doing good things. Now there's other bacteria that cause problems. You don't want them in. But as long as we have a healthy, we call it a microflora. What is growing inside of us is a healthy microflora. Makes you feel much better. That's why probiotics and things like that, which help us to digest right. All right. Metabolic diversity gives prokaryotes the ability to live almost anywhere there is a source of energy and carbon. They're everywhere. Prokaryotes harvest energy and nutrients in 4 different ways. I'm not going to ask you each of these ways. What I want you to know from this slide is photoautotrophs. They can absorb sunlight and make their own food. Chemoautotrophs. They can use the bond energy in different substances like sulfur, and they can break those bonds and make their own food. There is photoheterotrophs, chemoheterotrophs, where you can use the power of the sun to break down everything. So these guys are everywhere. They can feed in all these different ways. That's what makes bacteria so successful. They can live everywhere. Many bacteria, most archaeans, are anaerobes. Remember what that means. They can live where there's no oxygen. Remember when they don't have a mitochondria, so when they break down sugars, they don't use oxygen. They get 2 ATP per glucose unit, but that's enough for a single cell to survive on. Some can tolerate an oxygen-free environment. Others are obligate anaerobes, where they need different conditions at different times. So this is just a slide to tell you they can feed everywhere. All right. They're small, Bacteria come in three basic shapes, and we name the bacteria according to the shape. Coccus is sphere, little round, and you all know that from bacteria like Staphylococcus, which causes strep throat. There's a Bacillus, which is a rod-shaped, and Spirillum, which is a spiral-shaped bacteria. All right, they're very small. It says the head of a pen. I believe that, well, it could be a felt pen. It looks more like a pencil head to me. That looks more like the ridges that would be in the lead, but could be a felt tip pen. But look how small those bacteria are. Those are Bacillus bacteria. You see little rod-shaped bacteria all over that pen or pencil tip. So what are they made of? They have a nucleoid, not a nucleus. Remember, there's no membrane-bound nucleus. The nucleoid is where the circular chromosome hangs out, and usually it's not curled up as a chromosome, so it's just hanging out there so that it can be unzipped and copies made so that proteins can be made. They have a cell wall which surrounds the plasma membrane, and it has that slimy layer on the outside. Remember, we've already talked about that capsule. And that capsule, that slimy layer, helps them to stick to other things, helps them to stick to each other. They have a flagella, which rotate like propellers, an amazing flagellum. They have that pili, which extends from the cell surface for adhesion or motion. So those little hair-like things, and it helps them to stick, and then they can move those, and they can like crawl along the surface. And remember, one of those can turn into a sex pili, which will they can swap plasmids back and forth, just little strands of DNA. So there's the three shapes up there again. Here is our basic body plan. We've already gone over that in the previous chapter. Know the basic components of a bacteria. All right, so traits that bacteria and the archaea share. There's no nuclear envelope. They have no nucleus, generally a singular chromosome. They have a cell wall in most species. They do have ribosomes, so they do have this organelle that will make proteins, and that's distributed out. They reproduce asexually by binary fission. They just not break into, but they make a copy of the DNA that goes to the opposite end. We'll see a slide on that. And the capacity for gene exchange among existing cells by conjugation, transduction, transformation. We'll talk about those here just in a minute. So there's that bacteria body plan with the flagellum, the pili. Here is the DNA hanging out in the nucleoid region. Cytoplasm, which has ribosomes, it will have lysosomes there also. All right, bacteria and archaea reproduce by binary fission. That's what we're talking about. The DNA replicates, and the parent cell divides in two. This is the way your cells divide. Mitosis, where you just make a copy, they divide in two, and you now have two identical copies. And so growth for us is through mitosis. As we replace skin cells, it's through mitosis. The only place in our bodies where meiosis is taking place is in the sex organs, where you have half the genetic materials. You split the chromosome pairs apart to make a sperm. They also engage in this horizontal gene transfer, in which one individual requires genes from another of the same species or a different species. So there is different ways of horizontal gene transfer. All right, there's the DNA inside of a bacteria. Why that one's so square, I don't know. But there's the DNA, and notice what happens as it's getting ready to divide. It'll replicate. They've shown you two different colors, but it's an exact copy of the DNA, which is good. So the chromosome, it now has two chromosomes, and what's going to happen is now they're going to go out to different parts of the cell, and then the cytoplasm will pinch in half, and now you've got two of them which are identical to the one at the first. Don't let this purple chromosome throw you off. It's exactly the same one. They just showed you two colors to show how they're splitting apart from each other. So what about these horizontal gene transfers? Conjugation, the transfer of a plasmid between two bacteria with that sex pilus. So the pili attach to each other, and they swap the plasmid back and forth. No big deal. Transduction is the transfer of genes through bacteria, through viruses. Transformation. Genes acquired from the environment. This one is scary. Scientists have done some things with this that are kind of alarming. This is where a bacteria dies, the body breaks apart, the DNA is laying out, a new bacteria comes over and is absorbing things to feed on, and it'll pick up that DNA and incorporate it in its own DNA, and it changes. That's transformation. So there's two bacteria with the sex pilus. I don't know why this one only has the one pili with this picture. I don't know. That's the way it is. Here is conjugation, where they will have a conjugation tube and they'll swap a little bit of material. All right, this is transformation. Look at this guy. I'm not going to ask you this guy. This guy's frightening to me. Frederick Griffin changed Streptococcus bacteria from harmless to deadly by transformation. He mixed harmless cells with dead cells of a harmful strain. DNA from the harmful strain was picked up and used by the harmless bacteria, and it made it different, transformed it into something bad. This is what scientists are doing for governments to make biological weapons. We don't have antibodies for these particular ones, and so yeah, they can be nasty. All right. Bacteria are the oldest, most diverse, and most abundant prokaryotic lineage. Most are harmless or benefit us by releasing oxygen, good. Fixing nitrogen, also good. Or cycling nutrients. Plants especially need nitrogen to grow, and as they grow, they capture the sunlight, turn it into chemical bonds and sugars. So fixing nitrogen is key. So bacteria can do that, which is great. The bacteria will actually be around the plant roots, will fix nitrogen. So they're in a symbiotic relationship. So they fix the nitrogen that the plant needs. And to keep the bacteria there, the roots will actually start secreting sugars for the bacteria to eat. And so it keeps the bacteria there. The bacteria fix nitrogen. So they're helping each other out, which is a great relationship. But some bacteria are pathogens that cause diseases in humans. Thermophiles are some that can live in very hot conditions. They have a heat-stable DNA polymerase. Don't worry about that. That's how they can survive in these hot conditions. Here's our cyanobacteria. This is the blue-green algae, cyanobacteria. But they have these special heterocysts that can fix nitrogen in the water systems. Great. We need nitrogen in the water systems. They fix it, so they do a really good service of nitrogen fixation for our water. I'm not going to ask you the different groups of bacteria. If you take microbiology, and I suggest you do, you'll learn so much about bacteria and viruses and how we can, deal with these things and be safe ourselves. But I'm not going to ask you these. Just know there's different types of bacteria. There's these proteobacteria. There's gram-positive, which just means they have a different cell wall that accepts a stain. So some form endospores. Now know this for the test. Bacteria can form endospores. When conditions become unfavorable, they can germinate in the human body to make deadly toxins such as anthrax, tetanus, and botulism toxins. So what is an endospore? It's almost like a seed from a plant. It's got that tough seed coat on it. An endospore will actually form around the DNA of the bacteria, and it can survive harsh conditions until things get favorable again, and then it can open up and start to live again. But when it forms an endospore, we can actually make it into a powder, which is what anthrax, botulism toxins. Botulism is food poisoning, and some bright bulb discovered if you inject food poisoning bacteria in your skin, it causes wrinkles to disappear because it swells. Ooh, but some people like to do that. That's different strokes for different folks, I guess. Spire cleats, they're spring-shaped. I'm not going to ask you about these. This is what causes Lyme disease, by the way. It's carried in ticks. Chlamydia, there you go. Sexually transmitted disease is in this spirochete chlamydia, so there you go. So here's examples of disease-causing bacteria: whooping cough, gonorrhea, sexually transmitted, Rocky Mountain spotted fever. What's the vector? It is ticks, and here's cholera, which is a diarrheal illness. Then we have the different types of bacteria and the diseases they cause. I'm not going to ask you these and the diseases they cause. Just know that there's diseases that these bacteria cause. There's tetanus, strep throat, Lyme disease, syphilis, tuberculosis. So, all right. Now, the Archaeans. We're getting close to the end of this chapter. This has been a long chapter just because of all the COVID-19 talk. They were recently discovered. They are the third domain. They live everywhere. Many live in very hot or very salty. I mean, that's why they can live everywhere. These guys can adapt to boiling water. They can live in that. They can live in ice. They in Antarctica. They can live in the Dead Sea. They called that the Dead Sea because they didn't think anything was living in there. No, they live in the Dead Sea. But hardly any of these cause human disease. Why are they a new domain? Because scientists have discovered through the genomes that they are more closely related to eukaryotes than the bacteria are, so they just separated them out. So how did they discover these? They were comparing ribosomal RNAs among bacteria species, and Carl Woese discovered that some fell into a distinct group. Just realize they are more closely related to the eukaryotes than the bacteria, is why they're in their own domain. Some are extreme thermophiles. They love heat. They can grow in hot springs, like at Yellowstone National Park. And we got a picture of one of the hot springs. And, also, the Archaeans are very typically colorful as they grow on things. Some are extreme halophiles. They love salt, so they can live in the Dead Sea and the Great Salt Lake. And so here's a spring, and you can see the Archaeans growing on that. Very hot, but there they are. They are doing well. This is the Great Salt Lake in Utah, but they are growing there also. That is not an Archaean. That's a dude with weird yellow glasses on. I don't know why he's there. Anyway, some are metagens. They produce methane. So they're chemoautotropes that form ATP by pulling electrons from hydrogen gases, or acetate forming methane. Just realize the Archaeans in extreme environments, and they can get energy in those environments. Some live in sewage, marsh sediments. Realize marsh sediments, that's where there's no oxygen at all. Animal gut, again, all are strictly anaerobic there. All right. I love this picture. That is just an odd picture, isn't it? What are they focused on? There we go. They live inside the gut of a cow as they go from stomach to stomach, and they produce methane, which is released from the backside of the cow and goes up. If you have heard about the Green New Deal, where they want to get rid of everything that's causing pollutants, carbon dioxide increase in our atmosphere, they want to get rid of cows because they do this. So anyway, goodbye steaks and hamburgers and everything else. All right, that's the end of this chapter. I know a lot to cover, but an important chapter, especially with what we're going through right now. Watch this video when you get a chance, and look for the next chapter on the protists coming soon.